Welcome. Today's lesson is on genetics for beginners. We're going to demonstrate today how to use a Punnett square so that we can predict the genotypes of various crosses we encounter. The Punnett square was actually developed by Reginald C. Punnett. He developed it so that scientists could use it to keep track of all the alleles as they're tracking these genotypes. An allele is kind of a piece or a part of the gene that's on the chromosome that will allow us to kind of see or illustrate where that trait is. So the allele is that coded part of the gene that the trait that we're studying. And we put a letter arbitrarily by it so that we can keep track of it. So let's take a look at one example. The first example I want to show you is one when we have a heterozygous and we cross it with another heterozygous. And these R's represent rounded or wrinkled seed. So the first one I want to show you is, here's one individual and here's the other individual, kind of the mother and the father, if you will. In plants, we're talking about pollen and coming from the anther and the stamen, and then of course the female part is the ovary and the pistil and the stigma. So anyway, what we're going to do here is we're going to take a look at two alleles. One is for rounded seed, and then another one is for wrinkled seed. So little r is wrinkled seed. I'm just going to abbreviate it. And big R is rounded. So if we take a look at pollen, it's going to produce two different kinds of alleles, a big R and a little r. So these alleles will be located in the gametes of the pollen. So here is basically one pollen or sperm cell right here, and here's another one. And then of course on the other side we're going to be looking at two alleles that will be in the gametes of the mother, or the ovum, which will eventually turn into the seed. So that's big R and little r. So if this big R from the pollen fertilizes the big R from the ovary, that seed is going to be big R, and if we were to plant that seed, it would grow into a seed that will be rounded. So that would be the phenotype. On this side, we'd have little r, big R, would be a heterozygous. Obviously, the big R is going to be from the ovary, and the little r is going to be from the pollen. Then over here, this pollen grain, if it fertilizes that ovum, we're going to get a seed that's going to have a genotype of big R, little r. And if this pollen grain fertilizes that ovum, it's going to be little r, little r. So if we're going to break this problem down, you're going to notice that if we look at the genotypes only now, you're going to have big R. That's going to be one type, so that would be this individual right here in this quadrant. And then we have right here in these two quadrants, we have big R, little r. And over in this quadrant, we have little r, little r. So those are the different kinds of genotypes that this particular cross will bring. So we'll bring one big R, two little r's, big R's, here and here. And then over here, lastly, we have one little r, little r. So if we were to put some percentages behind it, the big R is 25%. The big R is 50%. And then the little r is 25%. Now, another way of looking at this is if we take this a little bit further and we look at just the phenotypes, in other words, the physical characteristics. Like, for instance, how many of these offspring are going to produce rounded seed? Well, we know that, of course, big R is dominant. So if we take a look at I'm going to change color here. If we take a look at these three quadrants, they're going to be producing the seed that's going to be rounded. So what percentage of that would be? That's 25, 25, and 25. That's 75%, and that's going to be rounded. And then if we look at right here in this quadrant, it's going to be 25% wrinkled. So in this particular case, in this cross, heterozygous, we get a 75-25. Or another way to look at it is a ratio. How many rounds we have? We have 3. And how many wrinkled? We have 1. So it's a 3-to-1 ratio. So one of the things you can deduce is that a heterozygous cross of the heterozygous is always going to get you a 3-to-1 ratio. And that'll be helpful in some of the other problems that we have down the road. So if you kind of remember, heterozygous means a 3-to-1 ratio, 75-25. That'll help you a lot and actually expedite some of your problem solving. A word problem, because this one's probably word problems are probably the most difficult to look at, but I'd like to share with you a kind of a strategy that I like to use. The skin of tomatoes can be smooth or hairy. In other words, 2 kinds of textures, one smooth, big S, or hairy, little s. That means smooth is the dominant trait. Those varieties are called ponderosa, and they're smooth. They're big S, and versus hairy, which are peach varieties, are little s, little s. So I hope you got that. There are 2 kinds, smooth and hairy. Big S is the dominant one, or the smooth, and the recessive is hairy. Then when we go to crossing those 2, we take the ponderosa, the smooth textured one, and cross it with a peach, both from a parental generation. You cross those, this is what the F1 produces. And this is what I would like you to underline when you're doing this problem. Let's go back one. 520 smooth and 174 hairy. What? Those ratios are really important because it's those ratios that's going to determine the inheritance. What kind of ratio is that? If you don't know what it is, just take 520, divide it by 174, and it's going to come out to about a 3.1. In other words, the smooth are 3 times as many as the hairy. Does that sound familiar? It should, because we talked about that at the very first one. Heterozygous cross with a heterozygous cross produces, go ahead and do this, produces a 3-to-1 ratio. And that's what this is. But we didn't start off with that. What we started with were 2 purebreds. In this case, a smooth or ponderosa type, big S, right? That's the only kind of gamete it can produce. And the hairy one can only produce little s. Let me do that again. So it's going to be a big S and then a little s right there. There we go. So when the pollen grain fertilizes the ovule, you're going to get seeds that have big S, little s. And they're always going to be smooth. All right? So just like this word problem says, the cross of the 2, which I just did right here, always produce smooth. That's how I know smooth is dominant. When we do all of that, what's going to happen is, now we're going to take these individuals, and I could put another S here, and another small s there, and then we can fill out the rest of it. But no matter what happens, if you take the parental strain, just like Gregory Mendel did, and cross them, you're always going to get the dominant trait coming out. But that also means that all of the offspring are going to be heterozygous. So we're going to take 2 of these, this one, and cross them. So what are the gametes possible right here? Well, it's going to be big S, little s. Big S, little s. So when this pollen grain fertilizes that ovum, we're going to get big S, big S. Over here, we're going to get big S, little s. Over here, the same thing, big S, little s. And then here, when this small s, pollen grain fertilizes that ovum, little s, little s. So what are you going to get? Just like we thought we might, This is a 3-to-1 ratio. How many smooth over here? 1, 2, 3. And we have 1 hairy. Just like this. So that's how we got that particular result, was because we took the parental strain, crossed the 2 parental strains, then took 2 of the F1s out, in other words, big S, little s, crossed those 2, heterozygous on heterozygous, and got a 3-to-1 ratio. You see, if you understood the first problem and all 3 steps that Gregory Mendel does, this will be the end of part 1 of Genetics for Beginners. Please see the video on part 2 for Genetics for Beginners. We'll be talking about X-linked traits, also how to predict crosses in co and incomplete dominance. I think you'll want to see that. Also, the 2 other videos you should look at, if you're a little confused at this particular point, watch the Lingo for Genetics and or Mendelian Genetics, and that'll help kind of score away some of the possibly the language or jargon terms. So remember, this is the end of number 1. Continue with part 2. Thanks.